Okay, can somebody please confirm that you can hear me?
Yes.
Great.
Yep,
I can hear you.
Picture correct, but
I can hear you.
I didn't have time to do it, but I think I can use this today. So let's go ahead and plug this in. Are there any questions before I get started?
Yes. Do we need to memorize the names of the people who made the inventions?
Can you repeat that? For some reason you were breaking up on me.
Do we need to remember the name of the people and the year of the inventions?
Oh, when they're talking about like the history of microbiology,
Yes.
on the important events, and I'll go over most of those, you do need to know the people who were involved in those important events. But on the dates, there's only one date I'm going to ask you to remember.
Okay.
And that's the first fossil records that we have in the, well, the fossil of bacteria.
Amen.
So I'll discuss that in the slide. Let me see if this works. All right, can you hear me now?
Yes.
And is the clarity a little better?
Yes.
All right, great. Let's see, let me load that and let's go on. So we haven't come to the names and the dates, but when we're talking about the history of microbiology, there will be a large number of names and dates. And except for one date, you don't need to know the dates. All right. Any other questions? If not, let's go ahead and get started. I'm having a hard time finding my two here. There we go. So I believe last time we had talked about that you can find microbial growth, meaning microbes or bacteria and other microbes in all of these locations, with the exception of lava. And I thought I had my syllabus out here. I don't see it. So let me get that. I'll just like to show you the schedule before we begin. Essentially, we're going to be covering chapter 1 and 10 today in the lecture, and we won't finish. Up a little further up. So right here. And we'll also be in the lab covering lab 01. Remember lab 00, the worksheet for the lab has to be turned in by 8 a.m. Friday, this Friday, April 8th, or you're going to be dropped. And I've got something going on. I think I have a dental appointment, so I'll probably check that early in the morning. So do get it turned in by at least 8 a.m. Any questions about what we're doing? All right, I'll leave that up for the lab. Close that. And move on to the next slide. Actually, I should clear this. So I'll be ready for next time. But we'll move on and I'll just save it. Where is everywhere? When we're saying that microbes are everywhere, then they are in most environments, on land and in the water. And I mentioned then they're in the air, so you can find them in the atmosphere, up about a mile. You can find them subsurface on the land and under the ocean floor. You can find microbes on all plants and animals. So you can find them essentially wherever liquid water is present, and temperatures between a -20 degrees Celsius and about 120 degrees Celsius. Remember, 100 degrees Celsius is boiling water, so they can survive above boiling water, at least some of them. And we do have them below a -20, but they're not metabolically active, and they will be frozen below a -20. And you'd say, well, listen, zero freezing. Well, some microbes, as well as some other organisms, can make a chemical antifreeze. And that's why some microbes can survive and be metabolically active at temperatures down to about a -20. Any question about that? I'm talking about Celsius, by the way, degrees Celsius.
Dr. Volk,
Uh-huh.
are you showing us a PowerPoint right now?
I'm
I was about to
supposed to
ask the
be,
same
am I not
question.
doing that?
No, we
No.
can't say anything.
All right, let me get that going. Yeah, whenever I show you the schedule, if you're not seeing it, let me know. You should be able to see it now.
Yeah, I can see it now. Thank you.
The trouble
that's
is
better.
on my end, without, the only place I know that this is recording and showing is this little red. Actually, that means it's recording. I don't even know how I tell that this is sharing the screen, other than the stop share. I guess that could tell me. Sorry about that. Let's move forward. Microbes are numerically the most abundant life form on the planet Earth. How many microorganisms are present? When we're looking at 1 gram of soil, we can find about 10 to the 8th prokaryotic cells, meaning bacteria and archaea. I don't know if that refers to single-celled eukaryotes, but let's go with prokaryotes. And if you were to count the viruses in that, as well as prions, it would be higher than this. In fact, it would be about 10 times higher than that. But we're not going to look at viruses and prions because, well, they're special. They're not cellular. And it depends on what type of soil you have for exactly how many you can find. Like if you were to take soil, 1 gram of soil from the Sahara Desert, you'd find less than 10 to the 8th cells in it, living cells. And that's because the soil of the Sahara Desert isn't very nutritious and there's not very much water. And the wetter and the more organic material you have in the soil, the more living cells you can find. But on average, you'll get 10 to the 8th cells per gram of soil. In a milliliter of ocean water, and that's 1 gram of ocean water, do you think you get more or less cells than you'd find in soil?
More?
More.
Say more.
Anyone else? That's a hint that that's not correct. Ocean water, the oceans, excuse me, I'm struggling with my coat because I have the window open. It's such a nice day and it's a little chilly in here. And I could turn on the heat, but I decided it's a nice warm day, so I didn't turn on the heat. But the oceans are really a desert location on the planet Earth. So you find fewer living cells in 1 gram or 1 milliliter of ocean water. How about 1 gram of human feces? You find more or fewer cells than you find in 1 gram of soil.
More.
Yeah, you do find more. Human feces is generally fairly wet and it has lots of organic matter in it. So you find lots of cells in it, plus the fact that we have a bunch of cells in our intestine that helps us digest our food. And actually about half of the weight, and I don't know if that's dry weight or wet weight or what, but about half of the feces weight is actually from microorganisms. On one human being, do you have more or fewer cells?
More?
You have more because a human weighs much more than 1 gram. And we have lots of microorganisms on our skin and in our intestines and inside of us. And of course, on the entire earth, you have more than 1 gram of soil. So that one's pretty obvious. And here we're looking at the numbers. 1 milliliter of ocean water actually only has about 10 to the 6 prokaryotes. And that's less than 1 gram of soil, having about 10 to the 8. In a human feces, if it's fresh, you got about 10 to the 11th microorganisms in it. However, if it's soil that's in the desert, not soil, a feces that's in the desert and it's dried out, you'll only have about 10 to the 6th living cells in it. But generally, when we talk about human feces, we're talking about fresh feces. And so we'll say 10 to the 11th. And there, I got it there. 55% of the wet weight of human feces is actually microbes. On one human, we have about 3.8 times 10 to the 13 microorganisms. And that's on our skin, in our hair, the eyeballs, the lips, in the gastrointestinal tract, and a few other places in the body. We have lots of microbes all over the human body. On the entire earth, the human population, it's almost 8 billion now. It's between 7 and 8 billion. And then we look at the number of microorganisms on the planet Earth. We have 1 nonillion. That is 1 times 10 to the 10 power 30, meaning 1 followed by 30 zeros. And most of you have probably never even heard of a nonillion, right? So we have lots of prokaryotic organisms on the planet Earth. And don't ask me how they get this estimate. It's an estimate, because nobody's ever gone and counted the number of microbes on the planet Earth. Same with the number of microbes on a person. When we're talking about microorganisms, you should realize that some microbes cause human disease. And these microorganisms, we call them pathogenic microbes. So a microbe that causes disease is a pathogenic microbe. Realize that most microorganisms do not cause disease. Only a small percentage of the total microorganisms on the planet are pathogenic or capable of causing disease. When we're talking about disease, we will make the distinction between an infectious disease and an inherited disease. An infectious disease is a disease caused by a microorganism or a parasitic worm. And that's why it's infectious, because it can spread from one patient to another. What are some examples of an infectious disease?
C. diff.
Say that again.
C. diff.
C. diff. Yep. C. diff. Clostridium difficile. What's another example of an infectious disease?
The flu.
Yeah, the flu, caused by influenza, the influenza virus. And what's an inherited disease?
Genetically,
Yeah,
genetic.
it's a genetic disease, one that you're born with. And what's an example of an inherited disease? Nobody can give an example of an inherited disease? Most cases of Tay-Sachs diseases are inherited. Sickle cell anemia is a case of an inherited disease, meaning the child will be born with an inherited disease. An infectious disease is only one that somebody gets when they are infected with a microbe that causes the disease. Like C. diff, you only get that when you're exposed to the bacterium C. diff, which is Clostridium difficile. And if you get it on your skin, you don't get an infection, but if you get it internally, you get C. diff, the disease, which I guess would be called gastroenteritis. All right. Oh, let me mention in this class, we'll largely be talking about infectious diseases because they are caused by microorganisms. Here's a picture of some microbes. Let me blow this up a little. You can see they come in various shapes and sizes. There's 1 micrometer. This looks like it could be E. coli, which is a little bit larger than most bacteria because it's a little longer. The width is about the same size as bacteria, but most prokaryotic cells are about 1 micrometer. And I guess that would be in diameter. Here is some fungi, different shapes. That actually looks to me like a white blood cell, but it's some cell that moves by phagocytosis. This is a microbe, but it's colonial. And you see each dot here, let me blow this up really big. Each dot is a cell. So this is the parent organism, and it is a microbe because you can't see it, at least not very well. You might be able to see this as a dot with your unaided eye. So we look at it under a microscope. And this organism has a bunch of babies. It has 1, 2, 3, 4, 5, 6 baby colonies. And this will open up, releasing the babies to go out, and then they will colonize some other area. Or if this is water, they'll just float around some other place. This is actually Volvox, if you studied that in your Biology 160 class. And I think that next one is another, I was thinking it was another yeast, but actually that might be, that's a white blood cell, a lymphocyte. I'm not sure what that is. Oh, HIV. Which is a microbe, but it's a virus. Oh, that's about right. Let's go on. We shrink that just a little. Microorganisms do play beneficial roles. We mentioned that they help us decompose organic waste, and actually almost, not quite all organic waste is decomposed by microorganisms. You can have some fungi, which could be like mushrooms, decomposing organic waste. And there's actually some plants that are not green, so they're not photosynthetic, that are parasitic, and they can help decompose organic waste. But generally, organic waste is decomposed by microorganisms and fungi. Some microorganisms are producers in the ecosystem, and they make us food, and that would be the photosynthetic microorganisms, or the green prokaryotes, although Many of them aren't green in color, but some of them are, as well as the chemosynthetic prokaryotic cells. They are producers in the ecosystem, and they can make glucose from CO2 and water. Both the photosynthetic and the chemosynthetic organisms can do that. And so we call them producers. We mentioned that microorganisms are very important in the nitrogen cycle. Without them, nitrogen would not recycle on the planet. We can use microorganisms to make certain industrial chemicals like ethanol and acetone. We use microbes to make insecticides. And this is sort of important because we sometimes want to use a insecticide that's naturally produced, not man-made. And these insecticides usually are less toxic to us than a man-made insecticide. We use microorganisms to make actually we use them to make all fermented foods, such as vinegar, most cheeses, all leavened bread, and then beer. And we'll talk a little bit more about fermented foods a little bit later in the term. But we get them from microorganisms. Microorganisms make products used in manufacturing, like cellulase. We use microorganisms to make antibiotics, like penicillin G. We don't make penicillin G in the lab, meaning it's not man-made. We actually grow it, and then a microbe makes the penicillin G antibiotics. There's many others that we grow as well. And then other products used in medicine, like today, insulin is made in a microorganism. It's true that humans make insulin, and my father was a diabetic and he first took insulin. And when he first took it, his only option of getting it was human insulin. And then they converted it to sheep insulin. And I don't remember what was after sheep insulin, but then they started growing it in microorganisms and then purify it. And today, because it's much cheaper to make that way, that's the way all insulin is made that I'm aware of, at least that's sold to the general public. Microorganisms are important in biotechnology and genetic engineering. Biotechnology is the use of microbes and organisms to produce foods and products for human use. Humans have been using biotechnology for centuries. Like the making of wine uses biotechnology. Any question about that? And we've been breeding corn using biotechnology to make it so that we can get either more corn oil from the corn or less corn oil from the corn. And we have corn now that you can get 80% of the weight in the corn being greater than 80% of the weight being oil. And I don't remember what the low number is. It's something like 10% or 5% of the weight of corn can be oil. And they're approaching the lower limit for the oil because corn needs some oil in it for the plant to grow. And we've approached that lower limit. Anyways, the point is biotechnology has been used for centuries. The new aspect of biotechnology is genetic engineering. And this is where we take a gene of interest out of some organism like a human, like the human gene for insulin, and then we put it into a microorganism. And that is called genetic engineering. Some applications of genetic engineering: we use it to produce a variety of proteins from bacteria and fungi. such as when we're making vaccines for certain enzymes. We use biotechnology for gene therapy, such as when a child is born with a defective immunosuppressive gene, not immunosuppressive, immune functioning gene, meaning they don't have a complete immune system. Usually it's you're missing the ability to make an antibody. And you can put in the correct gene into their cells and then inject the cell into the child. And that we call gene therapy, where the child is getting the correct gene to make up for the defective gene. Any questions about any of that? And then genetically modified crops is an application of genetic engineering, such as genetically modified bacteria. I already gave one example when we put the human insulin gene in bacteria like E. coli. That's genetic engineering. Another example, which I don't think it's on the market anymore, but you might remember the flavor saver tomato. It was a tomato that you could have fully ripe for a long period of time, so the stores liked them, meaning that the tomato didn't quickly go bad in the store. And they called it the flavor saver tomato. Apparently not enough people bought them because I don't see the flavor saver tomato in the stores anymore. But that was a genetically modified crop. Any question about any of that? All right. Microbial ecology is the ecology that microbes do. We already mentioned that microbes are important in the ecology of the planet Earth because they're important in recycling nitrogen. Well, microbes are also important in recycling carbon, recycling sulfur, and phosphorus. So there is a carbon cycle, a sulfur cycle, and a phosphorus cycle. And it's similar to the nitrogen cycle, where these elements are recycling on the planet. The difference is that the nitrogen cycle largely runs because of microbes, whereas the sulfur cycle, it does run because of microbes and certain steps, but there are some steps, many steps of the sulfur cycle that happen without the use of microbes. Any question about that? Like when sulfur is on a rock, you can have water dissolve out the sulfur, or you can have erosion, wind or rain or some other water can remove the sulfur, and then it'll be dissolved in the water. And I'm not real good on knowing the sulfur cycle where it happens, but you can have a microbe change it from sulfate to hydrogen sulfide gas, and then it can go up into the air. You can have another microbe pull down hydrogen sulfide gas and convert it to some other form, which would probably be organic sulfur. And that's the sulfur that we use as humans, an organic source of sulfur. Like sulfate to us, we can't use it. I don't think it's toxic to us really. It's probably not very tasty. But hydrogen sulfide gas would be a toxic form of sulfur to us. And so we get our sulfur from the food we or drink, the food we eat or the drink we drink. Is that the right way to say that? Some examples of sulfur that we get is protein has some sulfur in it, methionine, and mushrooms and eggs would be where, like humans, oops, didn't mean to move that, where humans get sulfur. The nutrients are recycled on the planet Earth by microbes. When something dies, it'll be either decayed so that the nutrients can recycle and be reused by something else, and it'll be recycled by either a microbe or by a fungi. The fungi, which are not microbes, are largely mushrooms. So those are some examples of the microbially mediated biochemical cycles that support life on Earth. And that's the carbon cycle, the nitrogen cycle, the nutrient cycle, the sulfur cycle, and the phosphorus cycle. Certain steps in all of these cycles are done by microorganisms. And in the nitrogen cycle, almost every step is done by a microorganism. The first people to understand the microbial mediated biochemical cycles on the planet Earth were studied by Winogradsky and Bajorink in the 1880s to 1892. And you don't need to know the years, but you should know that Winogradsky led the effort of studying the microbial mediated biochemical cycles on the planet Earth. And they were the first to show that microorganisms did this cycling of nutrients. One reason why they were the first to see this is most microbiologists, even today, when they're studying microorganisms, they study them by isolating one species of microorganism and then studying that one species. Winogradsky and Bajorink did not do that. They studied microbes in mass where there are many species working together. And that's what happens in the nitrogen cycle. One species does one step, and then the next species does another step of the nitrogen cycle. And so that's why they were able to study these microbial mediated biochemical cycles. And they were not studying one species of microbe at a time. They were looking at a whole community of microbes. And most people don't do that even today. But to study the microbial mediated biochemical cycles, you need to study microbes in a community. All right, any question about that? So know that Winogradsky studied the recycling of nutrients on the planet Earth, but you don't need to know the years. Microbes are important to humans, like in bioremediation. Bioremediation literally means life and to remedy. We use living organisms to deal with an environmental problem. That is bioremediation, such as this oil spill here. They're spraying microbes on it so that the microbes will break down the oil, and they can break it down into carbon dioxide and water, removing the environmental problem, the oil spill. And as I said, we use that, bacteria and other microbes, to break down pollutants like an oil spill. We can use bacteria and other microbes to work on other pollutants like mercury. Bacteria can't break down mercury because it is an element, but it can convert it to a less toxic form. And then, as we mentioned, microbes are very important in the breaking down of sewage. But humans don't break down the sewage. We build the facility, and then we put the sewage in there, and then the microbes break down the sewage. All right, any question about bioremediation? I briefly just talked about this. Let me talk a little bit about it in a little more detail. We can use microbes to make biological insecticides. And this is important for human welfare because the microbes that make an insecticide, these insecticides are much less toxic to humans than a man-made insecticide. And so they're an alternative to a chemical pesticide. We can use microbes to prevent insect damage to crops, as well as prevent disease transmission. Some diseases are transmitted by insects. And if you have a biological insect on your crop, you'll help prevent not only damage to the crops from the insects, but you'll help prevent disease transmission. We're talking about plant diseases. An example is Bacillus thuringiensis. This bacteria is actually fatal to many insects, and it is harmless to humans and other animals and plants. And so you can put it on your plants and then protect the plant from many insects. An example of this was my father, way back in the 1960s, put Bacillus thuringiensis on his apple trees. And it did prevent the apples from getting worms. The problem is that my father was a little lazy and he only applied it one time. And you need to put it on the apples about every 2 weeks, which is the same thing you need to do with the chemical pesticide. You need to apply it multiple times on the apples. And he was putting it in Oregon, Western Oregon in the spring, which we get a lot of rain in Western Oregon in the spring. And the trouble with the Bacillus thuringiensis is the rain will wash it off. And so the apples did have worms, but that was because you needed to reapply it every 2 weeks, as well as apply it after every rain. All right, any questions about microbes making insecticides? Has anyone ever used Bacillus thuringiensis? I guess not. Actually, I've never used Bacillus thuringiensis myself, but my father used it. And if you're wondering, I was just a small child in the 1960s, and I somehow remember that. Not sure why. All right, let's change the subject if there's no questions. We're going to move on to the naming and classifying of microorganisms. And that was all started by this guy here, who both named all living things, as well as started the classification system. Does anybody know his name?
Linus?
Yeah, Linnaeus. Or his Scandinavian name, I don't know, I think it's Swedish. He liked Latin names so much that Linnaeus Latinized his own name. I think it's Linné, if you take it Swedish, and Carl Linné, but most people call him Carlus Linnaeus or Carl Linnaeus because he Latinized his own name. And he started both the scientific nomenclature as well as the classification scheme. He gave all organisms 2 names, their genus name and their specific epithet. So to name a species, it needs 2 names, the genus and the specific epithet. Like Homo sapiens, Homo is the genus Sapiens is the specific epithet, and the species is Homo sapien, which means wise man or humans. And we can call this the scientific nomenclature because this is the name we use in science. And it's also called the binomial nomenclature because all of the names have two names. Bi means two, nomal name, so two names. Or you could call it scientific binomial nomenclature. It's still used today. The scientific nomenclature that Carlos Linnaeus came up with, like he named humans Homo sapiens. His classification scheme is still in use, but it's been greatly modified. When he was classifying everything, he classified everything into an animal or a plant. And I would assume he classified microorganisms into plants. And today we don't classify things into animals and plants. We'll talk a little bit more about the classification system probably later today. I think I'll get to it. If not today, then tomorrow, or not tomorrow, our next class. So let's talk a little bit more about the nomenclature. This system is still used today. All names are either italicized or underlined. Like if I were to write it on the board, a scientific name, I would underline it because I can't write in italics. The genus name or the first name of the species name is always capitalized, and the specific epithet is always in lowercase. All the names are Latinized and they're used worldwide. So if ever you're using a scientific name, it will be understood in all countries in the world because we all use the same name to describe like humans, Homo sapien. And that's one advantage of being Latinized. We're all the same, regardless of what country you're in. The name can either be descriptive or honor a scientist. Like Homo sapien is descriptive. I think Homo also means walks on two legs or something like that. And then sapien, wise man. So Homo sapien, wise man. Escherichia coli is named in honor of a scientist. Esther was an early microbiologist, and Escherichia coli was named after that scientist. He was an early microbiologist. It's also descriptive. Coli is where we find E. coli, and that's found in the colon. So it's both honoring and descriptive. Staphylococcus aureus is a name that is descriptive. It literally means ball-shaped cells, coccus, that are golden, aureus, and the balls are in the shape of a grape cluster, staphylococcus, meaning little balls in the shape of a grape, cluster of grapes. Any question about any of that? The first time you use a scientific name, you must spell out the entire name. Like Escherichia coli should be used the first time you use it. But all other times you use the name, and we're talking about like in a paper or publication, something like that, or even when you're speaking, you should say the full name, Escherichia coli, the first time, and then all other times you refer to it, you can abbreviate by taking the first letter of the genus and then putting a period and then using the specific epithet, like E. coli is the abbreviation for Escherichia coli. For the abbreviation for Staphylococcus aureus, you say S. aureus. Any question about that? Both Staphylococcus aureus and E. coli are found in the human body. One name is so commonly used that even among scientists, they don't use spelling it out the first time. I'll just word it that way. Meaning you should, if you're being correct, say Escherichia coli the first time, and then all other times call it E. coli. But even among scientists, E. coli is so commonly used that everyone knows what you're talking about when you say E. coli. And so that's one case where people don't use Escherichia coli. They'll go E. coli the first time. But you shouldn't do it that way when you're being strictly correct, following the binomial nomenclature. All right, so let's turn on to classifying microorganisms. Before we can classify all living things, we need to talk about the 3-domain system, which is actually the first classification system we'll talk about. When we look at all living things, we can classify them into 3 domains. And a domain is also called the tree of life because it looks like a tree with 3 branches or 3 trunks. Okay? Which is kind of a funny looking tree when you think about it, but it branches like a tree. There's the main trunk, and then there will be branches off of it, and then branches off of that trunk after that branch. So there'll be smaller branches off of the main branch. And that's why we call it the tree of life. When we take a look at the 3-domain system, we find that there are 3 domains: the bacteria, the archaea, also called archaea bacteria, and the eukarya, such as plants and animals or eukaryotes. How the 3-domain system is defined is we simply sequence an organism, and its sequence tells us whether it is bacteria, archaea, or eukarya, meaning the sequence tells us which domain it goes into. So that when we sequence a bacteria, we'll find that the sequence is most similar to other bacteria, and it is different than the sequence that we find in archaea and different from the sequence that we find in eukarya. This initially was a shock to early biologists because we assumed the archaea and the bacteria were all prokaryotes. And before we did the sequencing, we actually lumped the archaea and the bacteria together, meaning we classified them together. But when we sequenced them, we realized the archaea are very distinct from the bacteria, and they are as different from the bacteria in their sequence of DNA than they are from the eukarya. In fact, if you look right here, oops, didn't quite fit that, shrink it down a little. If we look really closely here, oh, I see what it did. I lost the scroll on the left. Hopefully I'll get it back when I shrink it down. We can see that this branch here with the archaea and the eukarya actually was together for a short period of time. See, the archaea are actually a little bit more similar to the eukaryotes than they are similar to the bacteria. But that difference is very slight. The archaea are much different from the eukarya and the bacteria, meaning they're more similar to each other than they are to the eukarya. But when we look at how similar they are to the eukaryotes and the bacteria, the archaea are a little bit closer, just a little bit closer to the eukarya than they are to the bacteria. Any question about any of that? All right. The next breakdown after the domains is the kingdom. And with the eukaryotes, we can break them down into kingdom plants, the plantae, the kingdom animaliae, the animals, the kingdom fungi, the fungus, and kingdom protista, the protists. And we'll talk a little bit more about that later. I'm just saying that the plants and the animals belong to the domain Eukarya. When we look further at the domains, we note that they have two different cell types. And this is the way the author of your textbook calls them, a cell type. The domain bacteria has the cell type prokaryotes. The domain archaea has the cell type prokaryotes. The domain eukarya has the cell type eukaryotes. Some people, not your textbook author, call this division the division of the super kingdom. And actually the super kingdom was established long before the domain was established, meaning the domains came out in the 1970s when people first started sequencing living organisms. They realized that there were 3 domains. The cell type was discovered before that because we all knew that cells were either prokaryotes or eukaryotes. And I don't know when that was first came out, but it came out much earlier than the 70s. And somebody can look that up if you want. I don't know when they first came up with prokaryotes and eukaryotes, but it was a long time before the 1970s. We won't use the super kingdom because your textbook author calls it a cell type. Just know that prokaryotes is a cell type and eukaryotes is a cell type. And so we can lump the bacteria and the archaea into the same cell type because they're both prokaryotes, but they're different domains. The sequence of their DNA is very different from each other. All right, when we talk about the domain bacteria, the cell type is prokaryotes. The domain bacteria have peptidoglycan in their cell walls. So all of the bacteria that have a cell wall have peptidoglycan in their cell wall. And you should note that peptidoglycan is actually the major molecule in the cell wall of bacteria. Any question about any of that?
Is peptidoglycan a complex made with proteins and carbohydrates?
Yes, we will talk about that. Actually, peptidoglycan is a carbohydrate and a, not really a protein, but a short peptide. So we'll talk about that a little bit later. I'm going to turn this up because I had a hard time hearing you. So we'll talk a little bit more about peptidoglycan when we're talking about the cell wall. which I think is this lesson, but it might be the next lesson. The domain bacteria mostly divide or multiply by binary fission. That means when the cell splits into two, it will literally split into two. Binary fission. For energy use, the domain bacteria cells can get their energy needs by organic chemicals, such as glucose. And that's what we do to get our energy needs. We get our energy needs from organic chemical. But some of the domain bacteria can get their energy needs from an inorganic chemical, such as nitrate or hydrogen sulfide gas. And that would be the chemosynthetic bacteria. And then some of the domain bacteria can get their energy needs from photosynthesis. And that would be a photosynthetic bacteria. As you know, green plants are photosynthetic, and they get their energy needs whenever the light is out from photosynthesis. Most of the domain bacteria are free living decomposers or primary producers, meaning they either make food themselves, primary producers, or they decompose something. But some of the domain bacteria are parasitic. Some of them are pathogenic, meaning some of the domain bacteria, only a few of all of the domain bacteria are pathogenic, but a few are. And we'll study them because we're interested in the microorganisms that cause human disease. The domain archaea has a cell type prokaryotic. Does anybody know, do the archaea have peptidoglycan?
I don't think
Answer?
they do.
Say what?
I don't think so.
You're correct. The archaea have, most of them have cell walls, but even those that have cell walls do not have peptidoglycan. So peptidoglycan for our purposes is only found in domain bacteria. And I haven't talked about the eukaryotes yet, but you can kind of tell by the way I'm talking about peptidoglycan found only in the bacteria. We don't find peptidoglycan in eukaryotes. Some of the archaea are found in extreme environments, including the methanogens, the extreme halophiles, that means salt lover, the extreme thermophiles, those that live at very warm temperatures. I could actually say very hot temperatures, the thermophiles.
Is halophiles, isn't that salt loving?
Yes, halophile, salt loving. But when we look further, we can find the archaea in all of the Earth's biomes. They were initially found in the extreme environments because most other living things don't live in these extreme environments. But when we look at every biome on the planet, we can find archaea. In fact, they are a significant portion of the microbes in the soil, as well as in ocean water. They are a significant portion of the microorganisms in the soil and in ocean water. Does anybody know, or any of the domain archaea are pathogenic?
I don't think so.
You are correct. The archaea, none of them are pathogenic. They do not cause human disease. To our knowledge, there's no archaea that causes human disease. For this reason alone, after this lesson, we're not really going to talk about the archaea. because the main focus of this class is on microbes that cause human disease. And of the bacteria, or I should say of the living organisms, all of the living organisms that cause disease are either pathogenic bacteria or a pathogenic eukarya, meaning there are some disease-causing eukaryotic cells, and we'll talk a little bit about that. But like yeast infections, yeast is a eukaryote. If you come down with a worm infection, that is a worm, a helminth, but we could also say it is an animal or a, well, worms are animals. There are some pathogens which are protists as well, kingdom Protista. All right, any questions about the archaea? All right, the domain eukarya can be split into kingdoms, and we're going to use the 6-kingdom approach. And the domain eukarya gets 4 of the kingdoms. Kingdom Plantae, the plants, Kingdom Animalia, the animals, Kingdom Fungi, the fungus, Kingdom Protista, the protists. When we're splitting the domain this way, you should know that other people will use more than the kingdoms that we're putting out here. I haven't stated it, but because the domain bacteria and the domain archaea are not split into kingdoms, we can say they only have one kingdom, and that kingdom is the same name as the domain. So domain bacteria will also be kingdom bacteria. And domain archaea is also domain domain archaea is also kingdom archaea. Any question about that? So we'll use the 6-kingdom approach. There are different numbers of kingdoms. And in fact, biologists can't agree on how many kingdoms there are. Some people use the 3-kingdom approach, is that right? In which case each domain is also a kingdom. Other people use the 6-kingdom approach, and that you split the eukaryotes into plants, animals, fungi, and the protists. And I should mention the protist is a eukaryote, which is not a plant, animal, or fungi. So this Protista, that kingdom, is a collection of many different organisms which may or may not be related. And so we always knew that the kingdom Protista was not monophyletic. And some people will split the 6 kingdoms into more kingdoms. And what they'll do is they'll split the Protista into different numbers. Some people have the 9-kingdom approach, and you just split the Protista into, let's see, 6. So that would be like 3 or 4 kingdoms. And then the 15-kingdom approach uses the same thing. You just split the Protista into different kingdoms. You can get higher order kingdom, in which case you start splitting the bacteria and the archaea, as well as the eukaryotes. For example, you can split the plants. I used to know this. Some people say plants include the large kelp and the green plants. And you can split that into the green plants, and then the large kelp, the large algae. All right, any question about any of that? And here I state that even though many of them, like the plants, most of the fungi, some of the protista have cell walls, the animals do not have a cell wall. All of those cell walls lack the lack the molecule of peptidoglycan. So peptidoglycan, for our purposes, is only found in domain bacteria. And it's actually the main molecule of the cell wall of bacteria. Let's briefly talk about the kingdom fungi, singular fungus. They use an organic chemical for their energy needs. It's the same as us. We can split the fungi into the multicellular molds and mushrooms, meaning that's an organism composed of many different cells, like a mushroom, makes up a living organism that we call a mushroom. And we can also split the fungi into yeast. All of the yeasts are unicellular. Many of the yeast decomposers, so they decompose something, but some of the yeast are pathogens, such as, oh gosh, no, I just lost it. I had it on the tip of my tongue. Yeast infection caused by, oh no. Dropping it. Sorry, I'm just drawing a blank here. Does anyone know what the yeast infection is caused by? What species? I'll think about it and I'll get it to you in a little bit. I'm not getting the thing, the name. No, Saccharomyces cerevisiae is the yeast that makes bread. I can't remember. I'll think about it and get back to you on this. What causes a yeast infection is, I know it starts with a C, but I can't put the name together. All right, I'll work on it in my
Is
brain.
it Candy?
Yeah, Candida albicans. Thank you. I needed somebody to get started. I knew it started with a C, but I couldn't remember the name. Candida albicans causes most human yeast infections. We can split the kingdom Protista into different groups too, such as the algae, which are the plant-like protists, meaning they are photosynthetic producers. They make their own food from sunlight, CO2, and water. So they're photosynthetic. They make molecular oxygen. We can also split the Protista and the protozoa, like the paramecium, the euglena, the amoeba. All of these get their energy needs from some organic chemical, and they literally ingest that chemical, like glucose, to get their energy needs. And then there's a third of the Protista, and these are the fungus-like protists. So we have the plant-like protists, the algae, the protozoa, which I don't have that here, I should, the animal-like protists, and then there's the fungus-like protists. The point is that this kingdom is not monophyletic. The algae is very different in the sequence of DNA to the protozoa, which is different in its sequence of DNA to the fungus-like protist. One fungus-like protist is I think they call it water mold, which is a protist, not a fungus, that causes potato blight. And it was this fungus-like protus that ruined the Irish potato crop, which brought a bunch of the Irish over to America because they didn't have enough food to eat in Ireland. And anyways, you don't really need to know the fungus-like protus. All right, any question about the kingdom Protista, the kingdom Protus? They are eukaryotes, and we can divide the kingdom, the eukaryotes into kingdoms, such as the kingdom plants, the kingdom animals, the kingdom fungi, and the kingdom Protista. Realize that the kingdom does not have a definition. There is nothing that we can define an organism to state that it belongs to this kingdom. So the kingdom is, well, it's not defined, and biologists are currently arguing about what a kingdom is and how many kingdoms there are. And they've been arguing this for about 20 years now. So I'm going to guess that they're not going to ever resolve it. And it's just because kingdom is not defined. So different people have, well, I guess a different definition of what a kingdom is and how many there are. And like I said, we'll use the 6 kingdom approach where we have kingdom archaea, kingdom bacteria, and then we split the domain eukarya into 4 kingdoms: plants, animals, fungi, and then the protists. Of the kingdom plants, we can divide them into the mosses, the ferns, the conifers, the flowering plants. What you really need to know about kingdom plants is that all of the plants are multicellular. We don't have any plants which are unicellular, or meaning single cellular. So if you have a single cell organism, it's not a plant. It's either an archaebacteria or archaea, or a bacteria, or a fungus, or a protist. It's not a plant if it's unicellular. The same thing with the animal. All the animals are multicellular. There's no animal that's unicellular. We can divide the animals into sponges, the worms, which are helminths. We'll talk a little bit about them. The insects, and then the vertebrae, the vertebras. We're not going to talk about plants any further in this class because plants do not cause human disease. However, there are some animals which cause human disease, such as the worms. We'll talk about them. What comes under the talk of a microorganism? All right, bacteria is a microorganism. Archaea is a microorganism. Viruses and prions are microbes, but do realize they are not an organism because an organism has a cell, and viruses and prions do not have any cells. There are certain of the eukarya which are a microorganism. All of the protozoa are microorganisms. All of the molds, all of the yeast, and all of the yeast are single cell. Some of the algae are microorganisms. Generally, if the algae is unicellular, it's a microorganism. If, on the other hand, the algae is composed of many different cells, like the kelp that washes up on the Oregon and Washington beaches, then it is not a microorganism. And then the helminths fall under the category of a microorganism. And why are we considering the helminth a microorganism? Because most of the helminths, most of the worms are not microscopic, right? Like Tania saginata can extend from one end of our intestine to the other. So what the heck is that? Something like 30 meters long? Something like that. That's clearly not a microorganism, because if something's 30 meters long, you're not going to use the microscope to look at it, unless you're looking at the cells. Nobody knows why we call the worms a microorganism? All right, the helminths, the worms, are multicellular animals, but many of them are parasitic, such as the flatworms and the roundworms. So the helminths can cause human disease. Not all of them cause human disease. In fact, only a relatively few, meaning only a minor number of the helminths cause human disease. And those are the parasitic worms. Most of the worms which cause human disease are not microorganisms, at least when they are an adult. And there's the key there. But they are microorganisms when we look at the infective stage. Most of the helminths, when they are infective, they are microorganisms. So most of us, when we get a parasitic worm, we are consuming the fertilized egg of the worm. I guess that may not be true if you're consuming a cyst or a cyst, and we'll talk about that later. But most of the infective stage of the worms is a fertilized egg of the worm, and it is microscopic. But some of the life stages of the helminths, such as the adult, is not microscopic. And microbiologists study things that cause human disease, and some of the things that cause human disease are worms. And so that's one reason, as well as the fact that the infective stage for many of the worms is microscopic. And that's usually the fertilized egg of the worm. Any question about any of that? If not, let's talk a little bit about viruses. Oops, and then I've got to stop. I didn't realize it was time to stop. Viruses, they are acellular. They do have a genome, which is the core molecule of the virus, but that genome can be either DNA or RNA. Any questions about that? Viruses never have as their genome both DNA and RNA, it's one or the other. The core is surrounded by a protein coat. The coat may be surrounded or enclosed by a lipid envelope. And viruses only replicate when they are in their host cell. The viruses only replicate when they've infected their host cell. Outside of their host cell, a virus cannot replicate. Viruses are not part of the 3-domain system because they do not have cells. All right, any questions about viruses? We'll talk about viruses much later in the term, near the end of the term. Actually, I guess I can leave that there, because that's where we are. All right. If there's no questions, I'll see you in the lab at 6:30. Bye.